Log on, tune in, find out. Another good idea from Cambridge.
I have to. By the way, do you have your memories?
I put it on the computer.
So you know how to operate this computer. So better start.
He's prepared.
So, and title is very exciting. Experiences, and in plural. A lot of. In optimal design for population PK/PD modeling. And today's this session, it's actually a New Zealand session. I think I didn't know that they have plenty of statisticians. Usually New Zealand is known for other items, multiple items.
Why it's New Zealand?
Why New Zealand?
Tim was in Australia. New Zealand.
Where is Tim from?
My PhD advisor was from New Zealand, so maybe.
So, I concluded that he was graduated from Queensland University.
Which is in Australia.
It's in Australia.
Yes.
So you confused me completely.
Sit down.
But
Sit down.
he is
Sit down.
from pharmaceutical company, so they intentionally confuse people with information. So in any case, podium is yours.
Okay, so thank you for that.
So how long became the PhD student of Stephen?
Because he was working
He was in Queensland.
for
Oh,
10 years.
now you
because of-
confused me completely.
So thank you. Thank you all for being here on a Friday afternoon. Thanks especially to Basha and Sergey for inviting me and organizing this nice conference. By way of background, I'd just like to introduce myself as a recovering statistician. I did my PhD in optimal design with Steve Duffel and John Eccleston. And then I was lured away by Big Pharma, where I now work at Lilly in PK/PD. I'm actually in the pharmacometrics group, which is a new group that's just been formed within PK/PD. So there are quite a few statisticians at Lilly whose job title actually includes the word statistician, but I'm not one of those. My job title is research scientist. So I spend my days trying to pretend to be a scientist. So I get to do all kinds of fun things like predict human responses from animal data. I get to play with clinical trial data for new drugs and work on disease progression models and these kinds of things. So it's all very interesting scientifically, but in terms of statistical methods, it's not always that technically exciting. But I say I'm a recovering statistician because every now and then I'll relapse and I'll get to do some optimal design, which is what I'm going to talk about today. So this diagram is I've borrowed from a paper by some colleagues of mine from a few years ago, where they talk about the role of modeling and simulation throughout drug development. So the basic structure is you test your drugs in animals, so this is preclinical development, then go through phase 1 to phase 3, phase 3, and then post-registration, there might be more studies involved. So this diagram is actually about Lou Shiner's learn confirm paradigm. And what they actually do in the paper is just illustrate the role of PK/PD at each stage by giving some examples of what we do. So for example, in preclinical studies, we evaluate biomarkers, which is things that are easy to measure and it doesn't take too long to get some kind of response that are hopefully predictive of what happens later on in a patient. We try to predict human PK and PD from animal responses. And then in phase 1, once we get into human studies, for the first time we can start building our initial PK and PD models. We predict a response in the target population, because in phase 1 usually this is in healthy volunteers. Sometimes PK can change between healthy people and people who have the disease that we're trying to treat. In phase 2, we start studying a greater number of people, so we can look at responses in patients this time. So we can do things like build exposure-response relationships, so this relationship between drug concentration and the effect that the drug is having. We start to assess the impact of covariates. Covariates here means things like age, sex, weight. And then in phase 3, we get more and more patients. We can validate the PK/PD models that we've built in phase 2, look at need for dose adjustments in special populations, and so on. So these are just examples of some of the things that we do in PK/PD throughout the life of drug development. So I'm going to take an analogous approach in this talk and talk about the role of optimal design as it relates to all these types of things that we do in PK/PD. I'm going to skip this slide. This is just an overview of the structural sort of equations that we use, but this has been described very nicely this morning. I just wanted to have a few equations in my talk because there's not too many. So starting in preclinical studies, the role of optimal design. So in PK/PD in general, in preclinical studies, what we try to do is predict human response for certain doses, certain concentrations. So this starts with our animal studies where we test a number of animals at different dose levels and observe certain effects. And what we want to do is relate drug concentration to the drug effect. This can be efficacy or safety. A lot of the time we don't actually observe the concentrations in the same animals that we observe the effects in. So sometimes we just get a dose-response relationship and then have to take PK data from separate studies and merge them together. And so you get all kinds of different types of error introduced to the modeling here. But this is what we're trying to estimate, the relationship between concentration effect in animals, and we may assume that the same relationship holds in humans. That's not always the case. In fact, it's rarely ever the case. What we can do if we have some extra information on a similar drug in a similar target population, we might observe this kind of concentration effect response in humans. So we can look at how the animals and humans relate in terms of these relationships. Something else we do with animal data is try to predict human pharmacokinetics. So we use something like allometric scaling, where the body weight of the animals is related to one of the pharmacokinetic parameters, so clearance or volume. And usually we just use log body weight against log clearance or volume, and this has a linear relationship, which usually holds pretty well for most drugs. So once we have this model built for animals, then we can plug in a body weight for a human, say a 70 kilogram human, and predict what their PK parameter is. So, apparently we used to use 60 kilograms to get a typical human body weight, but that's increasing over the years. It's probably going to go up further still. So now we know about the relationship between concentration and effect, and from these models we know the relationship between dose and concentration, because we can put together a PK model to estimate that, to predict that concentration. I say we know these relationships, but really we're just predicting them. So we put these together and we get a relationship between dose and effect. So when we first move into our human studies, we know that we want to target a certain level of effect, so we know what kind of range of doses we want to start off doing these studies in. There is going to be a large amount of uncertainty around these relationships, partly because of the translation from animals to human, and we're also going to have error, obviously, in the animal studies themselves. So to give you an idea of just how much uncertainty there is here, in the allometric scaling, when we talk about comparing observed, say, clearance and predicted clearance from allometry, if you're within threefold of your prediction, so you're less than three times your prediction, or you're greater than 1/3 of your prediction, that's counted as a good prediction. If you're within tenfold of that, that's acceptable, but not as good as within threefold. So obviously with the dose predictions, there's a large range of uncertainty. So that's why in our phase 1 studies, we do a fairly large range of doses, so we can cover our bases. So let me talk about an example of an optimal design in preclinical studies. So for this particular example, we want to estimate this concentration effect relationship. Usually, like I said, we can't observe the concentrations in the same animals as we observe the effect. That's because usually these are in rats and mice, where it's really difficult to get more than one blood sample per animal, so there's no way you can really predict the PK profile very well. But in this particular example, we're testing drug X. I apologize, I'm going to refer to all of these as drug X. I assure you these are real examples for real drugs, but I'd have to have you all sign a confidentiality agreement if I told you what these were. So we're testing it for efficacy in mice over 4 weeks. There are 14 animals per dose group. QW means they're dosed once per week subcutaneously. And we're very lucky in that we're going to have a wealth of PK information from this study. We'll have 2 samples per animal. Usually we can take 1 sample at the end of the study, but We're also allowed to take one extra sample by retroorbital bleed, which means we take a sample from behind the eyeball, which doesn't sound very pleasant to me, which is why we can only take one extra sample like this. So this is a bit of a busy slide. This is the design that the biologist who was running this study set up. So the columns here are days of the week. The first dose is this green box here on a Monday, and then each subsequent green box is an extra dose. So there's one dose every week. The PK samples are given by these colors are a bit strange here, but different green color. So there's one PK, the pink boxes here are the PK samples. One day after the third dose, another PK sample 1 day after the 7th dose, and so on. And each animal is actually only getting one of these pink samples. So that's described in this table here. So animal 2001 will get one sample here, and then every animal gets a sample approximately 1 week after the final dose. So we have 14 animals per dose group. We're only using a subset of those for PK, but each of those animals is getting 2 samples, one 24 hours after the dose and one 1 week after the dose. So I said to the biologist, that's very nice, but next time just leave this to the professionals. We know what we're doing. So this is the model that we'd use. So we actually had previous data from a single dose PK study. So we know what the shape of the profile looks like after a single dose. We can use that to predict what's going to happen after 14 weeks of dosing. If you speak non-mem, then this might mean something to you, but really this is the important part, this set of differential equations. This is a subcutaneous model, so you have actually 3 compartments. There's this absorption compartment, a central compartment, and a peripheral compartment. So of course, because this is a model with 3 compartments, we call this a 2-compartment model. And you'll notice here this function of Cp. Cp is the concentration in plasma, and this is this Michaelis-Menten type of elimination. It's a nonlinear function of concentration, so we call this a nonlinear model. So in pharmacokinetics, all models are nonlinear in a statistical sense, but this one in particular is nonlinear because of this relationship in here. And this is some typical profiles for the 4 different dose groups that are going into this study after a single dose. So the black line here is the lowest dose, and then you've got increasing doses, and you can see because of that nonlinearity in the model, the shape of the curve changes depending on the dose. So if we plot this over 14 weeks, this is what the PK profile looks like according to that model. So each bump here is 1 week, so you have the drug is being absorbed into the body for a certain period, then it starts to be eliminated. And the numbers here represent the proposed sampling scheme. So this is 3 weeks, 7 weeks, 11 weeks, and the last samples there. So each number represents a different elementary design. So one animal, so there's 2 animals in this group here with 1 sample here and 1 sample there, and so on. So that was what we had to start with. So then I decided to use optimal design to try to optimize these sampling times. So I used WinPopt to do this. I just wanted to say a quick word about software. I tend to use WinPopt most of the time out of some kind of misplaced loyalty to my PhD advisor. We also use PFIM at Lilly. Some of these examples are in WinPopt, some are in PFIM. I'm fairly agnostic in terms of the software. I've tried some of the others as well. Franz will give a nice summary of this later, I'm sure. But in WinPopt, I use the option for an exchange algorithm to do the optimization rather than simulated annealing. And that's because this model is based on the solution to these ODEs. It takes quite a while to calculate a single information matrix because you need to calculate that several times to calculate derivatives and so on. So one calculation takes about 2 minutes. So to do this with simulated annealing, which needs thousands and thousands of those calculations, it would take forever. So I did a fairly simply constrained optimization problem with the exchange algorithm using a small discrete set of sampling times to choose from. And I left the times, so the time frame of the samples to be equivalent to what was in this original design. So 1 sample within this week, 1 sample within this week, and so on, and then 1 sample sometime after that last dose. Again, we can only take 2 samples per animal. And when we optimize this, you can see the sampling times move around a little bit. It's hard to tell with the scale of this, but you can see we're starting to cover a nicer range of the PK profile. So when you look at the output from WinPopped, the numbers in red here, which might be a bit hard to see, are the relative standard errors, and these are all very good. They're around 20% or lower for every parameter in the model. So it was a pretty good design. I highlighted on the bottom here. This took over 2 days to optimize. And this was using this fast exchange algorithm with only a small number of sampling times. So just for fun, I thought I'd put in the original proposed design, see what we'd get. And surprisingly, these standard errors are also all very good. They're all mostly around 20% or lower, one around 30% here, but this is actually a pretty decent design. And if I'd done this to begin with, it would have taken 2 minutes to evaluate, and it would have saved a couple of days of optimization. The efficiency of this design was about 80% of the original. So I think the lesson here for me, at least, was to not trust my intuition. Go with a biologist who probably knows what they're doing more than I do. And just check the design before you start looking at any kind of optimization. Which I think is an illustration of what Franz mentioned yesterday or the other day. It's hard to guess where these samples are really supposed to be when you have this kind of variability structure. This is probably an easy way to compare the relative standard errors between the two designs. Obviously, the optimized design gives you an improvement for each parameter, but overall, the original design is acceptable. Okay, so once we have our animal data and we've made some predictions about where the human dose is going to be, we go into our first human studies. And this is, as I said, over a wide range of doses. We give healthy volunteers usually a single dose of a drug. We do a dose escalation usually, so we give them a low dose, a group of people, and if they're okay, there's no safety or tolerability issues, and the next group comes in, we give them a higher dose, and so on. So in terms of PK, there's honestly not much role for optimal design here. We have a small number of subjects. They're kept in the clinic for a day or two, and we can take a large number of PK samples. So to calculate our PK parameters, we use what's called a conventional analysis or a non-compartmental analysis. So there's no mixed effects model involved at this stage. We do use the data to fit population PK models, but the main objective is to do this non-compartmental analysis. So just some rules of thumb for PK sampling in a phase 1 design. We can take a large number of samples, 12 to 18 at pre-specified times. And since these patients are in the clinic, we can really take these samples whenever we want within reason. So I've had one study where I asked for a PK sample around 2 or 3 in the morning. I wasn't very popular, but we did get the sample in time, and that gave us the opportunity to look at the distribution phase of that PK profile, which is good. Take a few samples in the absorption phase, a couple of samples around the maximum concentration, more in the distribution phase, and then in the terminal elimination phase, we take a few more samples. We try to sample up to 5 half-lives in the terminal phase, and that lets us estimate the half-life better. So, again, our prediction of the human PK at this stage has a lot of uncertainty. That's why we tend to use quite a lot of samples. For multiple dose studies, we also take samples at the first dose and the last dose, and then some trough samples in the last few days. So trough samples are concentrations just before the patient is given the drug. It helps us to make sure that they're actually taking the drug every day. This is just an illustration of where the sampling times might go for a typical PK model. So you have your absorption phase, Tmax, your elimination phase here. And we have, you can do a similar rule of thumb for PD models. So this is an Emax model. You want samples around the higher concentrations where you have the greatest effect, lower concentrations where you have the least effect, and sometime around your EC50, where you expect a 50% response. So like I said, we typically don't use optimal design for PK here, but there are some cases where we're collecting PD data where we can fit a mixed effects model, and so we can use optimal design. And here's one example of that. This is it's called a receptor occupancy study. Receptor occupancy gives you an idea of how well your drug is hitting the target. So it's a number between 0 and 100%, and that's described by this model here. So this is a function of C is the concentration. This is a typical Emacs model, and this is your PK model. This is a one-compartment model, so it has two compartments. This the absorption compartment isn't well. Sometimes it's included in these diagrams, sometimes it's not, which is I think why this is called a one-compartment model. So you have first-order absorption and elimination, and again we're taking intensive PK samples here, so we don't need to worry about fitting a pop PK model. All we're really worried about in the optimal design is estimating these parameters in our Emacs model. So this is what the PK model might look like. You see the concentration change over time. The PD model tells you how effect is related to concentration, but what you're really observing is this: your effect changes over time. So in the optimization of this study, we needed to determine the time points at which we measure this receptor occupancy, but we also need to take into account the dose levels, because if we give all of our doses at a high level, we're just going to get a whole lot of samples around here, which won't help us at all. Okay, so PK is estimated by conventional analysis, so we focus on PD. We have some flexibility in sample sizes and doses. This is not always the case, but for this particular study, we had some control over what doses are studied. This is just a few of the optimal design scenarios that I tried here. There are about 30 different scenarios that I tried. The reason was because we're optimizing dose levels as well as sampling times, which I haven't shown in here, and this is done in WinPopt, and as far as I know, there's no way of optimizing both at the same time. Maybe some of the other software can do that, but this is a fairly tedious process to do, so I set up a kind of a systematic combination of dose levels and time points, number of subjects per group, and so on. So it took a while, but eventually gave us a reasonable design for the study. So now we may move into phase 2. We're expanding our study sample size. We have a greater number of subjects. We're now in patients who have whatever disease we're trying to treat. We have some idea of what the PK looks like, and we're starting to measure effect in patients. So in this example that I'm showing, we have fewer PK samples per subject. So rather than those 12 to 18 samples from phase 1, we're limited, because the patients will only come in every now and then to have PK samples. They can't stay in the clinic overnight. Optimal PK sampling has already been determined for drug X. There's a different drug X now, based on a pop PK model built from rich phase 1 data. So when I say an optimal sampling scheme, like Valerie said yesterday, we don't actually use optimal designs. We find an optimal design, and then we look at the constraints and sampling windows and things like that, and end up with a design that is near enough to optimal. It's efficient enough for our purposes. So for this design, we calculated the optimal sampling times, constructed sampling windows around that, and that was fine for estimating this model. Drug X has been developed as a monotherapy in phase 1, which means it's just going to be given alone. There's no combination with other drugs. But prior to phase 2, the powers that be determined that we're now going to combine drug X with drug Y. Drug Y is already on the market. It's been shown to be very safe and effective, except for about 20% of the population. This drug does nothing or very little. So there are some patients, non-responders or partial responders to this drug, that drug Y does not work for. And it's hypothesized that drug X will help with the mechanism of drug Y to elucidate a response in these patients. So we also need to worry about now the PK for drug Y. Like I said, drug Y is already on the market, so we understand the PK of this very well. However, there is a potential for PK interaction between drug X and drug Y. So what does that mean? So drug Y is known to be metabolized primarily by a specific enzyme, and this is a cytochrome P450 that we heard about the other day. And drug X, unfortunately, is known to be a mild inhibitor of this enzyme. Drug X is fairly early in development, so we don't really understand the inhibition of this enzyme very well. We have some preclinical data which suggests that it may be an inhibitor of this enzyme, which means that if we give high enough doses of drug X, we're going to inhibit the enzyme, which means that drug Y is not going to be eliminated as fast as it usually is, which means the concentrations will be higher than they usually are. Maybe up to 30% based on our model so far. This is a potential safety issue for drug Y. So it's not good for patients because if their drug Y concentrations get high enough, there could be safety issues that they run into. Drug Y is known to be safe at normal concentrations that you'd see at that dose level, but you increase the concentrations, there's a greater probability for safety effects. So in terms of the study interpretation, this could also hurt us because we're testing the combination of drug X and drug Y and comparing that to drug Y alone. If we're increasing drug Y's concentration, we need to know that we're not just improving the effects of drug Y just by increasing the concentration. Because if that was the case, we would say, why develop this combination at all? We could just give drug Y at higher concentration. So at this point in drug development, going into a phase 2 study with a combination, you'd normally have a drug interaction study in a small number of patients. You do intensive PK sampling so you could compare the effect of one on the other. But because this decision was made fairly close to the start of the phase 2 study, we didn't want to spend time and money doing an extra study if we don't need to. So if this is say $1 billion drug by the time it gets to market, every day that you delay the release of this drug is potentially several $1 million of lost revenue. So it's very hard to take your time doing this kind of thing. So we didn't want to take time to do an extra drug interaction study. And I'm going to show how we're avoiding that. This is a diagram of the study design for phase 2. Drug Y is given for some time before randomization, and the effect of drug Y is measured before and after this treatment is given for a number of weeks. And this is to make sure that we actually have non-responders to the drug. So if they don't respond sufficiently after however many weeks, then we say, okay, they're really a non-responder. We'll randomize them to one of these treatment groups. A combination of the two drugs at some dose levels, drug Y alone or drug X alone. And then we'll compare the combination, the effect of the combination to the effect of drug Y alone, and we can see then whether the combination works any better than drug Y alone. And we'll also use this comparison to look at the differences in PK. So if the PK of drug Y in these patients is any different to the concentrations of drug Y in these patients, then we can conclude that there is some interaction. And we can also do the same for the drug X. We'll compare the combination to drug X alone. So using the optimal sampling scheme that we'd already developed, I used some simulations to determine whether we can actually pick up that type of change in PK concentrations between the two drugs. So, Franz actually brought to my attention a recent paper of hers about looking at using a criterion based on the world test, which I didn't use for this study. I just decided to use simulations. So I simulated about 200 replicate studies for 2 arms, the combination plus drug Y alone. And for each of those 200 replicates, I used a reduction in drug Y clearance of a certain level, 0 up to 50%, which corresponds to an increase in AUC of drug Y from 0 to 100%. Use the existing study design that was set up to test for an effect of drug Y. And using the simulated data, fit a population PK model to fit the model and estimate the AUC for each patient. This is all using non-mem, by the way. And for each of the 200 replicates, then we can do a test for a significant interaction. So we create a confidence interval of the ratio of AUCs, drug Y with and without drug X. And if that confidence interval is entirely within 80% to 125%, we conclude there's no significant interaction. So for each of the scenarios, we looked at the probability of that correct conclusion, and these are all pretty good. So if there's no actual effect of drug X on drug Y, then 91% of the time we'll make that correct conclusion based on this optimal design, which was designed solely for PK, for the PK model, not anything to do with this drug-drug interaction. If there is effect, even a small effect, so a 25% increase in AUC is really considered insignificant most of the time, because your between subject variability is going to be greater than that usually. If there's any kind of effect, we have a very high probability of detecting that. We did the same for the opposite comparison, drug Y and drug X compared to drug X alone. Again, we get high probabilities if there is some effect. If there's not an effect, if they're equivalent, our probability is a little bit lower, but this comparison was frankly not as important to us, because what we really wanted to know is whether the effect of drug Y is true rather than an interaction. So this is a separate, not as significant. So finally we go to phase 3. So we're looking at much larger studies. We're really more confirming than learning, especially in terms of PK. We know what our PK model does. We want to know whether we have an effect in a larger population. So for this particular optimal design, we had another drug X, and this is another combination. So this is an oncology study, which are quite often combinations given with 2 other drugs in this case in about 1,000 patients. We know that drug X may affect the PK of drug Y and or drug Z, so PK samples are going to be collected from a subset of those 1,000 patients, only 20 patients in each arm, and then we'll compare the PK. So this is a similar problem to what we had in phase 2. At the end of phase 2 meeting with the FDA, they looked at a sampling scheme that we had for all of these drugs, and we asked the FDA, will this sampling scheme be adequate to look at the effect of drug X on these other drugs? And they said, no, you need to take an extra couple of samples at 7 and 14 days for drug Z. The drug Z had a much longer half-life than the other drugs, so we were taking an extra sample at 3 weeks. They wanted us to take more at 1 and 2 weeks as well. We had a lot of earlier samples as well. So this was actually one of the first times that I'd done optimal design in my career at Lilly. So this is when I was fairly new. So I was known as a kind of PK sampling expert because I'd done some of this in my PhD. So they approached me and said, can you tell us whether this sampling is adequate? So I said, sure, I know how to do that. I used PFIM actually that time, plugged in the model, and this is the results I got. There's not that much difference between the 2 designs, so whether we use 7, 14 days or not, there's this obvious difference here. For the V2 parameter between subject variability, had a very large variance under the proposed design without the 2 extra samples, but we were not particularly interested in that. So we went back to the FDA and said, we don't need to do these samples because these are the results. Like I said, this is one of the first times I'd used optimal designs at Lilly. If I'd gone back and done this now, I would, I think, taken a completely different approach. For one thing, I ignored the fact that we're looking for a drug interaction. All I cared about here was looking at getting precise parameter estimates in the PK model. Another thing is we have a low number of patients, and they're having fairly intensive sampling. This was not going to be analyzed using a mixed effects model. This was just going to be a conventional PK analysis. So we didn't really need to do population PK design, but at that stage, that's all I really knew. And when you only have a hammer, everything looks like a nail. So the FDA came back and we changed our protocol to include these 2 samples. They didn't like this decision, obviously, and I can see why that is now, looking back at it. So, I mean, this phase 3 example is, I think, a little bit of a cop-out. It's not as exciting as, say, the model that Sebastian showed in the Alzheimer's disease. But it's the type of thing that we can look at in phase 3. So in summary, you can see how optimal design can be used throughout drug development in PK/PD. The current software tools, at least the ones that I've used, are very useful for the most part. However, just put together a short wish list. Maybe this can segue into the discussion later on. These are all fairly minor points, I think, but I wanted to include some things that I'd like to see in software, things that I don't really have the time to work on myself at the moment. But, like I mentioned, quite often we're looking at detecting drug interactions, and but that's sometimes secondary to actually fitting the PK model. So if there's a way of combining the criteria for parameter estimation and say the Wald test criterion, that would be nice. Flexibility in model structure. So I didn't really talk about this in the structure of the model. So we've seen this already this morning. We have quite often a combination of proportional and additive residual error. So I think at least as far as I can tell, in all the software, it's assumed that the standard deviation of the response is a linear function of the model response itself. I think that's right. So, but at Lilly, what we do is assume that the variance is a linear relationship of the square of the response. So that means you have, variances a times y squared, a times f squared plus b, whatever. So we often have to, restructure the model in non-mem, the residual error model structure to be able to use optimal design software. So it would be nice if we could do this either way, or maybe we should just do our residual error model differently at Lilly. Whether one is any better than the other, I'm sure some of you could tell me. PFIM does a nice plot of sampling times over your model once it's optimized them. Something like the plot that I showed you at the beginning with the preclinical design. That's just something nice. Winput doesn't happen to do at the moment. I like to use this as a kind of sanity test just to see where your samples line up with the curve of the model. Batch processing. So like in the example, the PD example where I showed you where I looked at a large number of combinations of doses and sampling times, it would be nice to be able to do this automatically somehow instead of a large number of manual intervention. This is a tough one, possibly parallel computing. I know PopEd, I think, does this, but to try to cut down the computation time of some of these optimizations would be nice. So, that was all I had for you, so thank you.
Okay, let us start with questions. Steven, do you think it's a good presentation?
Yeah. That's very good presentation.
Thank you.
To also, it's been nice to see the optimal design over such a broad range of different types of studies.
Yes.
I mean, irrespective of the tool you use, it looks good.
So, okay.
I think because they don't say it, but I think in PopEd now you can optimize both for dose and number of subjects and something
right?
done together. You don't have to do it in batch mode, no?
Yeah.
Okay, that's good to
You
know.
can have any model structure as you like.
Okay.
So, I mean, you linearize around the residual error as well.
Oh, very nice. Once I get it installed, I'll try that out. Thanks.
I just want to ask about your experience in Lilly, if you know about experiments in early phase or later phase which would be adaptive in nature, because this is a parallel what.
Yes. None of these are adaptive designs. We have actually a dedicated group for adaptive designs at the moment that I haven't been involved with yet. So I can't.
They do this. Do they use these methods which now appear a lot in statistical literature on adaptive designs, or they just do 3 pi plus 3 or something?
I couldn't tell you. I'm not sure what the differences are. Sorry.
Okay, that's one.
So in your first example, where the biologist came up with a design that was pretty good, and you said it's always worth looking at these and evaluating them, which obviously is a good general point, but in that case, is it really worth throwing away 20% of your data to save 2 days computing time?
Well, we're not necessarily throwing away 20% of the data. I mean, when you look at the predicted relative standard errors, by the way, I should say that once we get these predicted standard errors, we always evaluate them by simulation as well, because, you know, the different approximations will introduce error into those predictions. But because the standard errors were reasonable enough, the proposed design was so much more easier to do practically. It was, you know, I think it is worth saving 2 days of computation for that.
Okay. Any comments?
In what way was it easier?
The design?
Just having everything, was that the one with everything at the same time?
Yes.
Right?
Yes.
Oh, okay, that's fine. Thank
Okay.
It's easier for the biologists.
Because they thought of
you. You don't have serious mobile telephones going off.
Some questions. So, how to detect this interaction? Is it you call DDT?
DDI, drug-drug interaction.
It's actually drug-drug interaction. And how does it? Could it be in some sense equivalent just to minimizing variance of coefficient in the model, which is somehow describes
Right.
x1 times x2?
Right.
So there, I think technique is there. You just introduce criteria which puts extra weight on this parameter.
Right.
So
Right. Sure.
to some extent, it's done. Just
Is one parameter?
no, it's not. It could be, for instance, the following: you minimize the criterion, minimize determinant, given that variance of that particular parameter should be less than something, for instance.
Ah, okay.
It's doable, and it's known how to do this. And so.
When
Or you
people
could
will
combine two criteria. So if you
You wouldn't like to do a T-optimum design?
T-optimal?
To maximize the power?
No, Steve has worked on T-optimal designs, they don't work.
I already have some.
This must be a poor implementation. Is all.
No, can I modify that statement and say that
We will talk about T-optimal design during coffee or tea break. So okay, I think it's a very nice presentation. If there is no